Well, that's quite an act to follow. Pleasure to be here. This has just been a superb day, just very exciting to see folks that I hadn't seen in a long time. I used to work on liver biochemistry and toxicology years ago. I'm in the pulmonary division now, and I had no idea I would ever work on the liver again. But last year we did a strange thing, and that was that we wanted to do some mitochondrial experiments, and it's a lot easier to get mitochondria from the liver than it is from the lung, and so we decided to do the pilot studies on the liver mitochondria. And so I'm going to tell you really a work in progress on the work with cadmium in really emanating from that work on the effects in the mitochondria. So I wanted to start off with what has been the main focus of the lab for the past decade or so, and that is the development of high-resolution metabolomics, specifically toward environmental metabolomics. And so I want to just give a couple words on what I call the exposome and next-generation medicine. And this really emanates from the concept that Christopher Wilde introduced about a decade ago, that we needed to examine human exposures in the same way that we consider the genome, that we really needed to look at the sequence, the cumulative sequence of exposures during our lifetime. And so he termed this, coined this term the exposome, which he defined to include all of the exposures from conception onward. Recently, Gary Miller and I have pushed that forward a little bit in terms of definition, because if one really thinks about the concept, it's not just the exposures, but the impact of the exposures as well in terms of mutations and epigenetic effects. So just a minor modification, but it's conceptually the same, and that is that if we had the ability to sequence our exposures, then we really could complement the genome with the exposome. And the reason that this is so important, as we've already heard today, is that much of human disease is in fact determined by exposure. The concepts are now that the gene-environment interaction is probably far and away more important than just the genetic basis of disease. So the issue is how are we going to use it, how are we going to develop that, and how are we going to use it. And so we've been working on the concept of next-generation medicine. How could we do this? What we really need is something that's simple to implement, that's non-disruptive to contemporary standard of care, and effectively is cost-neutral because the health care is so expensive. And so the concept that we've been working on is that this could be achievable in terms of really just two major measurements. One is our genetic sequence, and of course we have the capability for doing that. It would cost about 1% of the healthcare dollars to do this over the next decade, and we could have the sequence for every American. So it's not unaffordable. But the other aspect of this, we would really need a much better way to capture the exposome. And so what the concept that I've been working on has been to develop an advanced blood chemistry approach. And the concept is that if we had the ability to measure 20,000 chemicals, or 20,000 metabolites, all of the products of the microbiome, our exposures, our drugs, the drug metabolites, and so forth. If we could measure 20,000 chemicals in the same way that we can measure 20,000 genes, then the combination of those two would give us 400 million parameters. That would give us sufficient definition that we could in fact define individuals in terms of their exposure and their health. So that's basically the concept that we're working on. We have now achieved the ability to measure 20,000 chemicals in a drop of blood. I think it's possible for us to take that upward to 1 million with existing technologies. We will need better data extraction techniques, and the informatics aspect is not quite there. But I think this is feasible, and that this will come, and this is how we're going to really be able to approach and take us to a new generation of medicine. So the issue has already been discussed as far as what we have to evaluate. We have the metabolome, which most people think about as the products of all of the enzymes and so forth that we have, our nutrients and enzymes, but that's really only a very small portion of what's in our bodies. We have, of course, many what are considered non-nutritive chemicals. Of course, the liver is very important in metabolizing these. We have, as we've already heard today, a large number of microbiome-related chemicals. We have a large number of chemicals from different dietary supplements, pharmaceuticals, but then the commercial products and the environmental chemicals, this is really quite remarkable. There's over 100,000 chemicals now registered with the EPA, over 10,000 in common use. So when we're talking about the exposome, we really have a very broad range of chemicals that we have to address. And so that is the main concept. So I've said enough on this. This is, I think, where we're going in the future. I think this is a realistic expectation that we'll have a much better way to deal with environmental-induced liver injury as we have these technologies developed. So now I want to talk about the topic that I was supposed to be talking about, and that is cadmium. And as I said, I haven't worked on liver in a long time, so you have to have to accept my apologies for being ignorant on this, because it's just something that you have to brush up on after a few decades. But as I said, what we were interested in was the question of basically why cadmium, which is not an oxidative metal, the ion does not undergo reversible oxidation reduction under biological conditions. Why should cadmium be an oxidant? Because you look, there's hundreds of studies on this. I mean, this is one of the most extensively studied toxic metals, metal ions. And so it causes oxidation, and so we were interested in that. We had done some experiments over a decade ago on the effect on, well, I guess about a decade ago, on thyroidoxin, where we found that if you put different metals into livers or into HeLa cells, among the most sensitive in oxidizing the mitochondrial thyroidoxin was cadmium. And so this was just really where we started. And the concept is that if we come back to the genome and the exposome, the concept is that we have all of these thiols and different proteins that really function as an interface between all of the environmental exposures and the function of the functional genome, the function of all that's encoded. And so the idea was that we would look at what the cadmium is doing, and specifically in the mitochondria. And so this jumped back. I apologize for where I am in the talk. I forgot that I put this in here. This is what we were doing on the lung. We had done redox proteomics on the lung, and what we found was that within a large number of proteins that are associated with the actin cytoskeleton, there are cysteines that are oxidized just if you add low-dose cadmium. Now, why this is a big deal is this is the amount of cadmium that you're going to eat in your salad tonight. It's not most of the cadmium research has been done on occupational levels of cadmium. What we're finding here is that if you just take the cadmium level that we eat in dinner and put that on cells in culture, it oxidizes cysteines and proteins, and it's not just a small effect. It's rather a large number of these. As you can see, all of the cysteines are here identified in red from these different proteins identified in yellow. And so it's really a whole network that is impacted in the cytoskeleton. So we worked on this quite a bit. So the idea was we wanted to move this forward in terms of looking at in the mitochondria. Now, what we found in the lung was that the cadmium activates pro-inflammatory and pro-fibrotic signaling. We found that the oxidation is actually due to inhibition of thyroidoxin reductase. This enzyme that Bert already told us is not important as far as keeping thyroidoxin reduced. But in any case, if you inhibit the thyroidoxin reductase, you do get oxidation of a lot of proteins, even if you can't see thyroidoxin oxidized all the time. And what this appears to do is causes a global shift in the protein. So there's a lot of proteins that are oxidized. And as I already indicated, the mitochondria are most sensitive. So that was what we wanted to look at. So the idea was we were going to just isolate mitochondria from the liver after we treated mice with cadmium. And we did this, and this is just more or less a standard type of method that had been done over decades. You see changes in the glutathione pool. You see oxidation of the glutathione. You see protein oxidation, loss of protein thiols. You see increased protein glutathione disulfides. So all of this is really expected from the literature. And so what we did was we used a mass spectrometry-based redox proteomics, which allows us to measure the percent oxidation of the different cysteines in different proteins. And so you can see that there's an overall shift in the oxidation. And when we use these, we used Metacore here, GeneGo software, to look at what pathways were impacted in the mitochondria. What was interesting was that the top two pathways here were branch chain amino acid metabolism. These are actually the dehydrogenases in the pathways. There were other pathways, many were significantly impacted by these, by the cadmium treatment. When we mapped this out, and I apologize for this slide, it didn't come out right, but when we mapped this out with the high-resolution metabolomics measurements on the mitochondria, what we found was that many of the proteins mapped to the same pathways with the metabolic changes. So in this case, these are all changes related to fatty acid metabolism, and it was really this association with fatty acid metabolism not really so evident in those top pathways from the GeneGo, but it was really this mapping of the metabolome to the redox proteome that got our attention, because this is really a disruption of the fatty acid metabolism. And so as we studied it more, we realized that there were several critical cysteines that were being oxidized in association with fatty acid metabolism. And this was what really got our attention in terms of the potential for fatty liver, that if you have the cadmium going into the mitochondria, inhibiting the thyroidoxin reductase, so now you were losing your ability to maintain your branch chain amino acid or branch chain fatty acid metabolism and your beta oxidation pathways, then you really could be contributing to fatty liver. And some of the specific metabolites, or a couple, there's some were diglycerides, some mapped to triglycerides. I don't have a lot of that data here, but this was showing even those first experiments that are already published are at a high dose cadmium. These are at much lower doses, and here in pathway analysis, and when you do all the corrections, you're seeing significance even at for a lot of these different diglycerides and triglycerides. This was a functional study. I'll just skip through this. It's already published. So we probably would have just moved on, because that was not our intent. Our intent was really just to take a look at could we combine the redox proteome and the metabolome data. But as we started to think about writing that article up, we ran across this paper that had just recently come out, and it was an epidemiologic study using NHANES data coming out of Johns Hopkins with cadmium exposure and liver disease from the NHANES data. And this, I'm not going to go through a lot of this, but one point that's really important here is that we don't have any good mechanism for humans do not have any good mechanism for getting rid of cadmium. And so we accumulate this over our lifetime. The actual population levels of cadmium have been decreasing in the United States due to decreasing smoking, because cigarette smoke has been the major source of cadmium. Most of our understanding of cadmium is at occupational exposure level or at smoking levels. What we're talking about in terms of the levels that we're studying now in the laboratory are, again, what we're seeing in our diet. And so we're taking this down to the level. And the reason this is important is that all the plants that are grown with lots of water, like our lettuce, well, how do plants get rid of cadmium? Well, they don't. They have vesicles, and they just pump that cadmium into the vesicles. And so the plants that we grow, they can tolerate the cadmium. Not that the cadmium isn't toxic to them, but they just have vesicles to sequester it so it doesn't kill them. That makes our vegetarian diets a delivery tool for a toxic species. And in fact, some of the fertilizers that are used are high cadmium content fertilizers. And so it is an issue, and I don't want to overemphasize that because we're very early in terms of thinking about this and doing the experimentation, but it is a cause for concern, and it is potentially controllable just by measuring the amount of cadmium in what you're putting on the fertilizer. But anyway, we accumulate it, and this study, I'm just going to highlight a couple things from the study. What they did was they just did a median value and looked at the high and the low. They found a cadmium increased risk of necroinflammation. They found increased NAFLD, and they found increased NASH associated with this is just ambient cadmium load. Now this is a little bit biased because in some of these calculations because there's smoking in there, but they have corrected for smoking, and the patterns still hold. And this is the kicker that they have buried in the discussion with regard to all-cause mortality. But buried in the discussion, they say the risk of liver-related mortality and high-exposure patients having more. Anyways, there's increased risk of liver-related mortality as well due to the cadmium. And again, this is what we are eating, so it's something we have to think about. So we've been studying low levels of cadmium, and I have some preliminary data from our first studies on this, and this was actually a lung study, so it wasn't designed to study liver, and we're funded to study the lung, but we decided we'd look at the liver anyway. And so it's the highest of what we considered a low dose. So this would be, in the top 10 percentile of what humans are exposed to approximately in this model. You see increases in metallothionein, as you would expect you should. You see non-significant increases in AST and ALT, but again, the numbers are small, so we don't know whether this will turn out to be significant when we look at larger numbers. I'm not experienced in liver pathology at all. This is just a oil red O stain. And this, if you look at the pictures, you can't believe this as far as it being different because we don't have enough samples, and it was very heterogeneous in terms of the pattern. But the important point is this was not a Western diet. This was the standard chow. So the fact that you're going to see anything here, just with this kind of a level it tells us that we need to look at this, and I don't have any problem with talking about it as far as unpublished data. I think others should look at this because this is of significant enough importance that I wouldn't want to rely upon my lab, even though I trust my people. I think it's something it has enough of a environmental and health impact that we need to look at it. And you can see the changes in different indicators, and I don't want to dwell on this. You can even see differences in the plasma, and I'm going to show this because we're seeing the same thing in humans. I'll jump through this to the end. So we ended up, as this past year, just scurrying around thinking about this. Wow, this is something of potential. It's interesting, but it's more of a concern than it is of interest, honestly. And so one of my colleagues at Emory, Robin Bostic, had a set of samples that he had in his freezer, and that they had measured urinary cadmium on the patients. And it was a study totally unrelated. They were studying prostate cancer, and they had samples from over 300 people. And so I said, hey, we'll run those on our metabolomics and just see what they look like. So we stratified them according to cadmium intake, and this was a metabolome-wide association study. So in this case, we're measuring about 16,000 metabolic features, and what you see is you have a huge false discovery rate penalty for so many comparisons, but still there are 39 differentially expressed metabolites here. And when we look at those, you see that in fact the individuals cluster pretty clearly. This is now just the top and the bottom quintile. You see they cluster pretty clearly between low cadmium and high cadmium classified by these metabolites. Now, those metabolites, most of these, when you're measuring 16,000 of them, you don't know what most of them are. So we just have been really exploring the data. You see if you do a principal component analysis on those, they separate fairly well, but not completely as far as the classification. But coming here to the end, what I wanted to talk about just briefly, these are 6 of the significant metabolites that are discriminating the high and the low cadmium exposures. Again, we don't have any information on liver, anything about the liver in these individuals. But it is interesting that that triglycerides and diglycerides do fall into the category. Hydroxynonenal, lipid peroxidation, is one of the discriminatory metabolites. These others, I think we need to take a minute and just really reflect on what we've heard today. Fatty liver, methionine choline deficient diet. That was one of the things we heard about. We heard about methylation of arsenic, another thing we heard about. We heard about methotrexate-induced liver injury. What more? There's been more that we've been hearing about today. But on this issue of one carbon metabolism, and I just think it's just too much of a coincidence that in a human population where we're classifying them just according to the cadmium intake, that we're popping up with these metabolic changes in the plasma of the triglycerides, diglycerides, and then the one carbon metabolism effect. So I think this is something we really do need to pay attention to as a community to really try to understand this. So where we are now is we're trying to look at it all. We can do the metabolomics, and we basically we get metabolites from 146 out of 154 metabolic pathways. We can do a transcriptome, and what we're finding is when we put transcriptome metabolome together and do what we call a TMWAS, that association, we can now scroll through the association, the strength of the associations or the significance of the associations, and we can take it down and isolate different metabolic clusters and gene expression clusters and bring it down to the point of being able to identify very specific dominant central hubs in terms of the interactions in terms of a toxicologic process. And what's really interesting about this is if you throw a third level on top of this, for instance, the redox proteome, you can now ask the question, which of these transcriptome metabolome networks are actually correlated with the mitochondria or with other organelles or the microbiome, for instance. And so I think this is this type of an interactive omic approach is really going to be very powerful in terms of answering some of these really difficult questions. So I think in terms of the title, I don't want you to get the impression that I'm selling this as fact. This is where we are in terms of thinking, in terms of the evidence. It's pretty clear that cadmium inhibits thyroidoxin reductase. It's pretty clear, at least in the lung, that at the environmental levels of cadmium that we have, that it is impacting the protein oxidation and the redox networks. It's pretty clear in the lung, at least, that it is impacting the cytoskeleton, that it is impacting pro-inflammatory and pro-fibrotic signaling. I don't know whether that's happening in the liver. We haven't done those studies. It's pretty clear in the liver that it does disrupt the mitochondrial branch chain amino acid and fatty acid metabolism. Lots of collaborators, and over the years, and I usually keep folks when I like Bert Watson's name on the list, because Bert really made seminal contributions when he was in my group, and it's wonderful to see him continuing and thriving here in Louisville. Most of the work on the cadmium has been done by Youngmi Go and Karan Uppal, long-term collaborators, Miriam Voss, Tom Ziegler, Arshad Kiami, and of course, for those from Louisville, Jesse Roman. So thank you.
Questions? Two-part question. As part of your metabolomic analysis, do you look at urate?
Yes, we get urate.
Urate is, I remember it's probably better than mine. You only looked at 150, but you remember whether it went up or down.
We can look at the data. I don't remember.
And the reason why I mentioned it, so the second thing is more of a comment. Those three metabolites, I think, are important in terms of purine metabolism.
Yes, that's purine metabolism. Yeah. Mm-hmm. Yeah, it's very, that is one thing that is important on all of the metabolomics and on these network maps. You don't, unless you look for it, you don't know whether things are going up or down. And in a metabolic sense, because if you inhibit a pathway, if something is upstream or downstream, you can have they can be correlated with changes, but actually going in the opposite direction. So that you really, to get at the mechanism, you really have to look at those details.
So one often thinks of cadmium and renal toxicity. In fact, chronic renal disease is somewhat not quite as bad as MASH, but it's something that's been increasing in prevalence as well. Have you done any metabolomic work on kidney tissue?
So not with the cadmium. That's interesting. I mean, because in reading the literature on the cadmium, the liver injury really precedes the kidney injury. And so, but what is remarkable is that if you get to my age, if you measure my liver, it's probably millimolar cadmium. I mean, it's an incredible, we have an incredible capacity to take up and maintain that cadmium. When the liver starts to decline, that's when the kidney gets hammered. Yeah.
So first of all, I never said anything about mitochondrial thyroid reductase.
Apologize.
But I was wondering, if you have such a huge cumulative body burden of cadmium, what difference does one more salad make?
Well, that's a good question. We have a tremendous capacity to accumulate it. I think the epidemiology is what we're going on. You don't know cause and effect in that, but that study is important because those were, that was from the NHANES data. As far as we know, the body burden is the best measure that we have of the total body burden is the urinary cadmium output. And so that's how they're measuring the body burden in terms of that urinary cadmium. Most of the cadmium is bound. Some of it's free. And we don't know much about the dynamics on that. If the reason that we looked at the thyroidoxin reductase was not something because we were smart, it was because I pulled out my inorganic chemistry book from college, the only college text I still have, inorganic chemistry. I loved that class. Didn't remember any of it, but I looked, opened up this book, cadmium. First paragraph on cadmium has a remarkable binding to selenium. So that was what we looked at. You go back through the literature, and the literature, they show that in mouse studies and rat studies, you can actually protect against cadmium toxicity with selenium. And so we looked at that, and then there's papers that glutathione peroxidases are inhibited by cadmium. And so it looks as though it really is an impact upon these antioxidant systems that are selenium dependent. And in fact, if you look at the cadmium burden in tissues, it's proportional. The amount of cadmium that we load in the liver and in the kidney is proportional to the amount of selenium in the liver and kidney. So I think that there's a story there that we really don't know, but you got to do those experiments too.
Another selenium protein is the T3, T4 synthesizing enzyme in the thyroid.
Mm-hmm.
And could some of these effects of cadmium be indirect via low T3 and low T4? Partial hypothyroidism?
Yeah, haven't thought about that. I think that's a good question. There's also a selenoprotein that's involved in that one of the methionine sulfoxide reductases is a selenoprotein, so that's another antioxidant protein. So, but that one is, I think that's really an important one that, and we can probably pick that out from our metabolomic studies because we do get T3 and T4 in there. Okay.